Good morning, dear friends. Question number 23 is on the screen. Consider the following statements. Statement A: the molecular weight of RNA polymerase is greater than that of DNA polymerase 1. Statement B: RNA polymerase starts chain without primer, whereas DNA polymerase 1 starts chain with primer. Which of the statements given above is or are correct? Option A, option B, option C, and option D. So, first of all, we understand the RNA polymerase and DNA polymerase. See the table. DNA polymerase in prokaryotes and eukaryotes. In prokaryotes, there are three types of DNA polymerase: polymerase 1, 2, and 3. And these are responsible to synthesize DNA on their template in 5 to 3 direction. These also having different types of activities, that is the exonuclease. Polymerase 1 having 5 to 3 prime or 3 to 5 prime, both type of exonuclease activity, and responsible for gap filling and DNA repair. Polymerase 2 having 3 to 5 prime exonuclease activity, and gap filling and DNA repair is their function. Polymerase 3 having both 5 to 3 prime or 3 to 5 prime exonuclease activity, and it is the main replication role involved in replication of the DNA. In eukaryotes, there are 5 types of DNA polymerase: polymerase alpha, beta, delta, gamma, omega. Alpha is responsible for replication of lagging strands in nucleus and works with delta polymerase. Polymerase beta जो है, it is responsible for gap filling and DNA repair in the nucleus. Delta is responsible for exonuclease activity and involved in replication of leading strand in nucleus, and it works with polymerase alpha. And then delta, this one, it having activity exonuclease in 3 to 5 prime, involved in replication in mitochondria. And last one, omega, having 3 to 5 direction exonuclease activity and involved in gap filling and DNA repair in nucleus. Prokaryotic RNA polymerase, consider the core enzyme consists of 5 evolutionary conjugate subunits: 2 alpha subunit, beta and beta dash, and omega. And these overall having total molecular weight of nearby 380,000 380 kilodalton. The task of transcribing nuclear gene is shared between 3 RNA polymerase in eukaryotes. These RNA polymerase are in prokaryotes. This one. In case of eukaryotes, there are 3 types of RNA polymerase: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. RNA polymerase 1 is responsible for the synthesis of large rRNA 28 and 18, 28S and 18S ribosomal rRNA. Polymerase 2 is responsible to synthesize mRNA, messenger RNA. In case of polymerase 3, it is responsible for the synthesis of tRNA and 5S rRNA. Now, if we see the DNA polymerase 1, these are present in prokaryotes. These are in prokaryotes. Polymerase 1, 2 and 3, as you see in table above. Polymerase 1 having 928 amino acid, and it is formed of single polypeptide chain, and its molecular weight is 109 kilodalton. Polymerase 2 having 783 amino acids. Polymerase 3, it is holoenzyme composed of nearby 10 proteins subunits. Main function of polymerase 1, that is DNA polymerase 1, having 5 to 3 prime exonuclease activity. It is popular as DNA repairing enzyme, and rather than DNA replication enzyme, has been have the ability to catalyze multiple polymerization prior to releasing the template DNA and connecting Okazaki fragments together by filling new DNA and removing RNA primers. Polymerase 2, in case of prokaryotes, having 3 to 5 prime exonuclease activity as well as 5 to 3 prime polymerase activities. It interacts with DNA polymerase 3 and responsible to maintain the fidelity and processivity of the DNA replication. It is responsible for proofreading, fidelity and processivity of newly formed DNA. Polymerase 3, it is responsible for DNA polymerization in 5 to 3 prime direction and add nearby 1000 nucleotide per second to the newly polymerized DNA strand. It also involved in proofreading and having exonuclease activity in 3 prime to 5 prime. That's all. From this discussion, we conclude that DNA, the molecular weight of RNA polymerase is greater than DNA polymerase. This one RNA polymerase weight, and this one DNA polymerase weight. ठीक है, it is 109 kilodalton of DNA polymerase 1, and in case of RNA polymerase 1, it is 380 kilodalton. That is, the RNA polymerase weight is higher than the DNA polymerase 1. Now, RNA primase starts chain with primer, whereas DNA polymerase 1 starts with primer. This is DNA. Now, undergo replication, transcription in case of RNA polymerase. This one RNA polymerase, and it starts to synthesize RNA. It may be rRNA, mRNA, and tRNA. It requires no RNA primer. In case of DNA polymerase 1, this is DNA, replications take place, fork formation. This one DNA polymerase. At the time of start, it requires RNA primer. In continuous strand at one time. In case of fragmented strand formation, that is formation Okazaki fragments, at each site it require RNA primer. Then statement B is also correct. RNA polymerase start chain without primer, and DNA polymerase 1 start chain with primer. These are primer. Then option C is correct. Same type of video I will come again. you see, like, share and comment. Thank you.